Agriculture Biotechnology from AAU, PhD in Biological Sciences from the University of Exeter, UK. I think she was working at that time in our department. She joined in 1991. 91 as an assistant professor. Then she received a Commonwealth Fellowship to do PhD in UK, and she went in 2004-5. 2001, and you came back in 2004. So her research focuses on addressing a few long-standing soil and water health issues due to abiding stressors on agricultural soils and groundwater of northeast India, such as low pH associated with aluminium, iron toxicity, nutrient deficiency, heavy metal contamination. She is keen to understand the microbiomes and the biological function and process through various omics approach towards harnessing the power of microorganisms to mitigate these issues. Really, she is going to talk about a little bit of transcriptomics that she did in micro. So that is also something that you can do in your research, the transcriptomic profile of microbes. A research group has isolated arsenic-tolerant bacteria from the contaminated groundwater of the sun and decipher the mechanism of arsenic transformation. The Metagenomic study reported the seasonal variation along with the impact of anthropogenic activities, water quality, bacterial community composition, the dynamics of Lothar Lake and largest freshwater lake in India. She has published more than 70 papers in several scientific journals, including Science. I think that came from her PhD work. Then BNC Microbiology +1. I think she is also associate editor of BNC Microbiology. Recently, she was appointed as associate editor of it. So I welcome Madhumita Madam to present her lecture. Yes, ma'am. Okay. Over to you.
Thank you, Sunita, for your kind introduction. And a very good afternoon now. I think you crossed it. So very good afternoon to all of you. So what I would like to be talking about, Sunita asked me to give you an advance, talk to you about advanced omics technology or advanced technology. So I assume your basic is clear. So you by now, at least even if it was not clear earlier, by now you must know what terms in biotechnology. Thank you. So I'll be talking on the bacterial physiology, as we are trying to understand specifically with relation to acid tolerance, how bacteria adapt to acid tolerance condition. Because in the northeast region we have soils which are acidic in nature. And because the soil is acidic in its nature, our productivity, agriculture productivity is low as compared to the national average. Okay. Then we see that many of the when there is heavy rainfall, we see many of our big trees, falling down. It's because the root system is not good. Okay, why the root system is not good? They are not strong enough to hold on to the ground because we have aluminium toxicity. Now, when they have acid soil, it results in aluminium toxicity and iron toxicity. So, let me briefly say about what that we have done. First, if you listen, let me just. So I'll be talking about the multi-omics as my difference suggested. I'll be talking about the multi-omics. So what is multi-omics? What is omics? You all know by now what omics is. It's a comprehensive or a global assessment of the set of molecules. Okay, these molecules can be DNA, they can be protein, they can be RNA, or they can be any metabolite. So when you're studying at the companies on a global level, then you say it's a omics. So this multi-omics, they comprise of genomics, they comprise of metagenomics, transcritomics, proteomics, and metabolomics. Yesterday, Robi said a lot about metabolomics. He talked about metabolomics. So today, I'm touching upon metagenomics and transcritomics. Okay, so the other possibility to understand, they offer possibility to understand and view biology from global perspective in a way that was not previously unthinkable. Previously, we used to isolate bacteria, look at their one, but at the large level, we did not know what was happening. So this omics has enabled us to understand how they function. They have three categories in multi-omics. That is, the first is the genome first, the phenotype first, and the environment first. Okay, so they all interact to produce, and depending upon the initial focus of investigation, what the focus of investigation is. You'll understand this more as I go through the slides. And this omics also helps to anticipate implementation in crop science to improve crop yields and enhance biotic and abiotic stress tolerance. I'll be talking about the abiotic stress tolerance. And they offer opportunity to achieve a further understanding of cells, organisms, and microbial communities, focusing on mechanisms involved in the growth, adaptation, development, disease progression, plant pathogen interaction, and plant health. So these are the possibilities that you can address with the help of multi-omics technology. So I'll start off with metagenomics. So what metagenomics is? It is the study, the taxonomic and functional composition of microbial communities from a phytomicrobe or agricultural. So, or it can be any other. It can be from any contaminated soil, it can be from water, it can be from anything. It can be from the human gut. You have microbiota from the gut, you must have heard about that, so everything. And you can perform this in two approaches. We have two approaches of performing this. One is the 16S RNA amplicon-based approach, and other is the shortcut metagenomic approach. Okay. Now, this metagenomics allows to discover new genes and proteins, or even the complete genomes of non-cultivated organisms in less time and with better accuracy than classical microbiology or molecular methods. Normally, when you're isolating bacteria or fungi or microorganisms from the soil, what we do is we make a culture medium, isn't it? And then we inoculate the substance and then we isolate the bacteria. Now, as you know, I think Dr. Dastagar also mentioned in 1 ml of this, you have billions of microbial cells in seawater. So similarly also in soil and in other places also, you have so many thousand billions of microbes, and all these microbes are not culturable. Why they are not culturable? Because we do not know what their requirements are, okay, what food they eat. So how will they grow? So we have not been able to understand, but with the metagenomics, because we are circumventing the cultural part, we can isolate them, or we can know, we are not really isolating, but we know the existence of those microbes. Okay? So that is why it is also said as a culture independent method. So this is how it is done. So you have the microbial community. You have this microbial community from any, it can be from any part. You can go either by RNA, 16S RNA gene profiling, just take the 16S and look at the profile, or you can go for the shotgun metagenomics. The 16S amplification, you got this, you get the amplification, you have the operational taxonomic unit generation, okay, and then you assemble them, and then you look at the abundance of the operational taxonomic unit. That means the bacteria, the families, what they are, okay. And this is how you get a profiling of this taxonomic profiling of the microbiota. In shotgun metagenomics, you fragment the DNA, sequence these fragments, okay. Then you have a metagenome read pools. You again take the help of bioinformatics, based analysis, okay. You do the taxonomic profiling here, or you can go for assembly-based analysis, that you assemble this sequences that you have got, okay. Then assemble and mapping. And then you can go for the functional analysis. So this is how the metagenome is done. Now these, before going to this, let me tell how we started. So we had this one time before where we reported the niche differentiation of below-ground microorganisms and the functional simulation Assam type tea soils. So you know, in Assam we grow tea, and tea soils are acidic in nature. Okay, so we wanted to understand what is the difference, what different levels, what are the microbes present in this tea soil. And we also looked at the diversity and functional properties of acid-tolerant bacteria isolated from tea plantation soils of Assam, because tea soils are acidic in nature, so we assume the bacteria which are present in these soils will be tolerant to the acidic condition. Now, I already mentioned, we are focusing on acid-tolerant genes or bacteria. So now we wanted to understand how management practice influence the microbial community structure in tea soils, because we already know that there are different tea soils, there are different, in different niches, you find different types of microbes. But as you know, in tea, you have a management practice where every December, November, December, you prune the tea so that you have a big table and it is easy for planting. So every year it's a must, okay? We wanted to understand what happens when you prune the soils, what happens in the soil, is there any change in the microbial community structure? Is there any change in the functional properties of this microbial microbes, or is there change in the aerobic factors in the soil? So we wanted to understand that. So what we do, we have an experimental tea garden here in the university, and there is a plot which is known as seed barley, okay? That those trees, they grow up to a tree. Actually, tea tree is a big tree, okay, going to 30 centimeters. But because of this cultural practices, we maintain a table so it's easy for planting. So at one plot receives pruning, is a management practice all the time, and another plot has never received pruning. Okay, so there is a difference in the management practice. So this is what indicates this. UP is unpruned and P is pruned. Okay, so we look at the metagenome of this and we find there is a difference. If we look at the abundance of these two different types of bacteria, there is a difference in unpruned and pruned. And then you can see this part, this type of bacteria, this is more in unpruned, there is less in this. And similarly for other bacteria also, you can see there is a difference. So as you can see from this metagenomic data, it was very clear to us that there is a difference between the management practices. And then we also looked at the relative abundance of dominant bacteria generated at UPM or P metagenome. So what is it? Dominant bacteria present, because we can see from this chart that some of these are dominant, they're equal, but then this is more dominant. Similarly, this part is more dominant than. So we wanted to understand better what is happening. So here again, this is pruned and this is unpruned. Yellow was unpruned. So you can see in the pruned soils, you find this bacillus. They are more compared to the unpruned soil. So this way, if you look closely, you find there are certain bacteria which are more abundant in the unpruned soil as compared to the pruned soil, or the vice versa. Okay. So this is a cog-based annotation of the predicted breeds in the UP and P metagenome. The value of the right shows the P values derived from two-tailed fishes, etc. This is basically looking at the diversity. This log looks at the diversity, and here it says what are the functional properties of the soils that of the bacteria that are abundant. So we find that there are some cell cycle control, cell division. Okay, they are almost equal. If you can see, they are almost equal. Then replication, recombination, repair are so similar, but there is a difference in general function prediction. Okay, so here you find that in pruned soil it is more as compared to the unpruned soil. So similarly again you find the lipid transport and metabolism is more in the unpruned than compared to the pruned soil. So this way you get an idea of what the function is. And this is also again showing the relative abundance of dominant PEG pathways. Okay, these are the pathways which operate in the soil, because the soil also has activity. So these again show that there is a difference. Suppose for ABC transporter, if you look at unpruned soil has more as compared to the pruned soil. Then if you look at here again somewhere, you find again a difference between this nitrogen metabolism is more, slightly more in compared in a pruned soil as compared to the unpruned soil. Then this table, this diagram is showing the enzymes associated with the two soil metagenomes. As you can see, this is the P, this is the unpruned soil. So hydrolysis enzymes are more active in the pruned soil. Okay. Then transferases are almost equal in both the soil samples. Isomers are also equal. Then ligase you find slightly more in the unpruned soil. And again, lyases are almost similar, whereas oxidoreductases are more in the unpruned soil. So along with the microbial population, there is also changes in the enzyme activity of the soil. So this is what I'm trying to tell you, that with the metagenome, it is possible to deduct this. Okay. Now, this is a comparative plot. This is a relationship between the most abundant genera of bacteria metabolism properties and their concerted influence on soil adaptive factors on the plant growth and development. So, from the two metagenome, we know that there are differences in the bacterial population and their abundance. Then, how do they relate actually? So, if you can look at this figure, you have this is the prune soil, okay, and this is the unpruned soil. So in unpruned soil, you have this dominance of Streptomyces, Pseudomonas, okay, whereas in the prune, Bacillus, if you remember, recollect the other slide that I showed, there also you can see the Bacillus was predominant. So these are some of the predominant bacteria found in the prune soil as compared with the unpruned soil. So when you look at the plants, tea plants, you find that the root length, dry mass, number of leaves, they are more in unpruned plots, okay, the trees growing in unpruned plots as compared to the pruned plots, that is the cultivated plots. So similarly in unpruned soil, what is happening? The acidic pH goes down, so you also have the aluminum saturation, which is not present in the unpruned soil. Sudipta, am I right? So this is all work of Sudipta sir. So if you need more information, you can ask him. Okay. So this is what we published. It was just published a couple of days back. So this is long-term pruning modulus microbial community structure and the function properties in the soil. It was published in Applied Soil Ecology, which is an impact of 4. And this is what I mentioned about. This is the plants growing in unpruned, and these are the plants growing in pruned. So you can see the stunted root is less, root development is less as compared to the unpruned soil. Okay. Okay, now we come to another part of work that we did. It is a seasonal variation of microbial community structure in the Lockton lake. You know, Lockton is the very famous lake with a lot of unique properties. So we wanted to know what is the variation in this microbial population during the different seasons. We wanted to, our hypothesis was that during different seasons, the microbial population, the dynamics will change. Okay. So what again we did the metagenome. So we took the samples from post-monsoon and winter, okay, and we looked at the microbial dynamics and the abundance. As you can see, there were differences. If you look at the color pattern, there were obviously differences between the post-monsoon and the winter collected samples. Now this plot, this slide is showing you the interaction, the environmental factors which influence the microbial interaction. Because seasonal variation will have changes in what? Main is temperature, okay? Then pH, then also other activity like this BOD, biological oxygen demand, the magnesium. So all these factors were taken into account to understand how these microbes interact with each other and how many microbes are involved in this process, okay? This is what it's showing. So if you look at the pH is an important factor here. So you can see that comparatively this many OTUs means just microbes, okay? So there are many microbes associated with this pH and they interact. So there is a lot of influence of pH on the interaction of the microbial population. And if you look at the temperature again, it's another factor which is very, now if you see and look at this, so in temperature you find 3 groups, okay? 3 groups, clusters here, you have different OTUs. They not only interact among themselves, but they are also interacting with the other groups. So a lot of influence of temperature on the interaction. Basically, this is trying to build a network to understand how the microbes network to survive on those conditions. Okay, is it clear? Am I clear? So this is the publication that we got from this bacterial operation taxonomic units. Replace the interactive roles of other operation taxonomic units under strong environmental changes. So if there is a strong environmental changes, there are changes in the microbial population, and some of them replace each other. Next we come, we work with soil, we work with water, we also work with food. Okay? Food is important. We have many fermented and ethnic foods in the northeast region. So this was towards understanding or developing a defined starter culture for us for ethnic rice-based alcohol production. I'm not sure how many of you know this, but rice-based alcoholic beverage, they're called by different names by different communities. One Haz is called by this Ahom communities, they call it as Haz. And these are some of the starter cultures, okay, that we collected from different communities. So this is a starter culture collected from the Sikh community, Diori community, Ahom, Dimasa, sorry. So all these ethnic communities make this, and they make this starter culture with, this is a starter culture, and this is the basis for the formation of the alcohol. So we wanted to understand, because this is the starter culture, this is the starting of everything. Okay, how your product will be. So we wanted to understand really what are the microbes present in the starter culture. Because ultimately it is the microbes which are, involved in all the process, either with saccharification of starch or production of alcohol. So we wanted to understand that. So this is again this one taxonomic clip of this different bacteria. So you find that about 28.8% sequence belong to the organism, because you have rice starch also in that starter culture. So you find rice also here, then 7.6 sequence belong to lactobacillus and 6.6 belong to the pseudomonas. And these are the different groups of bacteria and other stuff that you find in the starter culture. Now what we find from the starter culture, because we were interested in the fungal and the yeast flora associated in the starter culture, we find that there are fungi like Rhizopus, Meeker, Aspergillus. Okay, these are relatives found in most fermented foods in Southeast Asia. Southeast Asia has a lot of fermented foods, and this is commonly reported from many of the foods. Then muco, this is a potential biomass conversion. It converts the biomass because they have the cellulose activity, okay, amylase activity. And this is Aspergillus. This is again used in a for biomass conversion. Then among the yeast, you have this different yeast groups, okay, and these are this group of yeast, this Myrozyma, they are infected against spoilus fungi. Then Mucormyces, they are producer of acylated sphingolipids derivative. You have Saccharomyces, most effective ethanol producers. Everyone knows about that. Then you also find extremophile, osmotolerant, Demeromyces hansi. Then you find another group which are thermotolerant methylotrophic bacteria that produce methyl. And then you have spoilus yeast genus of. So all these organisms are present in the starter culture. Now this is what happens when you take the starter culture, because all these are there. So sometimes your product may not be safe. Okay, that is one thing. Because you see, they are also potential harmful bacteria. And our idea is to take efficient isolate. Why do we need so many isolates which produce off color? This they can produce off color. So sometimes when you have from different, the taste differs. Okay, the quality differs. It's because of the presence of all these microbial. Our idea was to isolate efficient microbes, okay, from this starter culture, so as to we could produce a defined starter culture for production of the house. Okay. So this study was important. In fact, this is one of the pioneering studies with this thing. We worked in 2014, but I think we published it quite late, 2018. But even now, this paper has a lot of citations and a lot of papers quote this paper, because this is one of the seminal papers. So this is what paper we did, is metagenomic analysis of microbial associated with traditional rice-based starter culture. This was published. Coming to next two. So that was all about metagenomics. So what we can do with metagenomics? It's with metagenomics you can study soil ecology, you can study soil, you can study to know the seasonal variation in water, in soil, and you can also apply metagenomics in food. Okay, transcritomics, I know you have already, you know what transcritomics is, so I'm not going to the detail. I know all of you know what transcritomics is. What I'm going to tell you is how we have applied transcritomics to identify genes and understand the bacterial physiology. So this is a walkthrough of transcritomics. So what we did, because as I mentioned, we were keen to understand how the bacteria responds to the acidic soil condition. So we had we grew in two conditions. One, was in the normal neutral condition at pH 7, and other was to go at pH 4.5. Okay, and then we extracted the total RNA, then we did all this fragmented RNA, we did the cDNA, prepared the cDNA. Okay, and then we went for sequencing, and we got this result. So we had the sequence information. So that was a transcriptome. Now from the transcriptome, we found that this is the expression of the genes at pH 7, and this is the expression of the genes at 4.5. So we find, what we find from it? Obviously there is a huge difference. Okay, some of the genes, especially in this region, you find that they are more expressed more as compared to this, and here they are, different sets of genes are expressed. So this clearly shows that when stress is applied, the bacteria respond differentially by expressing different genes. Okay, different condition they express, and that is how they are able to survive. So that also goes for others. That also goes for plants. That also goes for other things. But we were studying bacteria. So from this, from our study, we came to know that stress response. Okay, so you have for stress response, you have some general stress response proteins. They were upregulated in the acid stress condition. So this general stress proteins could be any proteins. It could be for heat. It could be for other biotic condition also. Any stress condition. So they were there. Then you had some proteins. You had some genes which are expressed or which are associated with osmotic stress. Now, when you have acidic condition, you also have osmotic stress, and in fact, you have proline. We found proline is a significant contributor in this. Then you also have oxidative stress. Okay. Then metabolic remodeling. The bacteria also remodel. Okay, themselves. They go for metabolic remodeling, like formative pathway, the regulators, and acidity alternatives. So they regulate this, and they also remodel the cell wall to cope with the acid resistance. Okay. So they have the membrane proteins, penicillin binding proteins. So these are some of the changes that we found. Then they also maintain the pure homeostasis to maintain the pH within the cell, because in the acidic condition it become acidic. So they don't want to have the acidic condition. They want to maintain a normal pH. So they have this, and they have this glutamate decarboxylase working in this. And so these were the three pathways that we found from the transcritomic data that are involved in helping the bacteria to cope with the acid resistance mechanism. So this is the paper that we published, the scientific reports. It adapts to acidic stress condition through a network of genes, insights from a genome transcriptomic analysis. Now we found, based on the data that we had, we found about 208 genes were expressed, okay, and about 70 genes were downregulated. So we took some of the genes. Now, just transcriptomic data will not help us. If you just say that it is there, it's not going to help us. We have to again prove and show. Okay, so what we did the qPCR, real-time PCR, which we talked about yesterday. Again, taking the condition, okay, in acidic condition and normal condition, and we looked at many of the genes. Some of the genes were annotated, we knew their function, but some of the genes that we found were expressed, overexpressed, had no annotation. We do not know what is the function of the genes. So as a scientist, it is our interest to know what the genes could be. Okay. So for this to extend this work, what we did is we already had some information. This was a paper that we published about acid tolerant bacterial amyloidification. It retains biocontrol efficiency. It is known as a biocontrol agent. Okay, this is Bacillus amyloidification. It is also a soil bacteria and resistance to acidic soil condition. And also, there is a pH-dependent gene expression which enables this bacteria to adapt to the acid stress condition. So we have this information in hand. Okay. Now, taking the information from the Bacillus megaterium, from the transcriptomic data, because transcriptomics, Metagenomics, all omics are big expensive, and as state universities, we are not in a position to do transcriptomics for all. So sometimes we have to take the data from others and see and apply them. Okay, so because Bacillus was same, Bacillus megaterium, Bacillus amyloidification, both acid tolerant, both belong to the Bacillus group. Okay, and And this Bacillus also, in addition to that, it also had biocontrol activity. So we wanted to take the help of transcritomic data from Megatherium and apply it to here. So we applied and we found that there are some genes which are upregulated and downregulated also during this process. And among this, we again did 35 genes in Bacillus amyloidifications. And one of the genes was interest to us was this BG 4142. It expressed 24-fold more in acidic condition as compared to the normal neutral condition. So we are very keen to understand and know what this gene is. Okay, till now we do not know. We just have the transcriptomic data. So what we did, we took the gene. So primer was designed for this. Okay, we knew in Bacillus migratorium that is a cupid containing domain. So we designed the primers. Okay, to perform an in silico by using amyloidifications 205 chromosome complete genome. Meanwhile, we were lucky that we also had the complete genome of amyloidifications. Okay, so we took the help of this data. Okay, and then the length of the sequence, it had about 308 sequences. Okay, we found. Then we took the DNA sequence, we amplified the product, then we sequenced the product, and this was the DNA sequence of the product. Okay. And we found that this gene ORF was located in the region of this much, okay, and annotated as QP domain containing protein. So this much information we had. Next, we wanted to know what this protein, what is the structure of this protein. So again, we took the help of bioinformatics. So Roman is here. If you need some more information, you can ask from Dr. Roman. So he has done all this part. So again, we had the DNA sequence. So from the DNA, we converted it into the amino acid sequence. Okay, so this was the amino acid sequence of that protein, and then taking help of this, a model was generated. Okay, now just generating a model will not do. You have to again test it whether it is accurate or not. So you go for Ramachandran plot. You do the Ramachandran plot here, and it was found that 96.2% of all the residues were in the favorite region. So anything above 85 is considered good. Okay, below 85 is good. And then 99.1% of all these residues were in the allowed limit. So this model was a fit model. So we took this as a base. And then we wanted to see how this interacts with other genes or other pathways, biochemical pathways. Okay, so this is the gene. It was annotated as BANU in this. It's been annotated as BANU in this genome sequence. So anyway, so this was our gene of interest. Now we took this gene of interest and we say how they interacted with others. Okay, so we find that it is interactive. If you can look at this, you can see this from database. This is from curated database, and this is experimentally proven. So this is not only taken for curated database, but also experimentally proven. So this was of much interest to us. Similarly, this was another gene. This is another gene which is interacted with other than also this gene. Because there are many other genes, it's not possible to do all. So we just took a few to just see initially. So this is of interest. Now this TUF, it is involved with the cell wall synthesis. You know, in gram-positive cell wall, you have teichoic acids and teichopuronic acids. Okay, this is what makes the cell wall. So we find that this, our product, the QP domain, the gene, interacts with this. It has an effect on this. So if you remove this gene, if you mutate this gene, it will affect this gene. That means the cell wall is affected. That means, although we do not know what the function of this gene was, we understand a little bit now that it might be involved with the cell wall. Okay, but simple understanding will not do. We have to again show experimentally. Okay. So this is about again how I'm not going into the detail of this, but this is again another gene, GLMS1, which works with the glutamate fructose 6-phosphate aminotransferase. So this is how it interacts with the formation of the cell wall component. Okay, and this is another gene which is involved with the maintaining this YPGA is involved with the maintaining the pH homeostasis within the cell. Okay, those 3 genes that I mentioned earlier. So this is a teichoic acid biosynthesis protein. It's an extracellular polysaccharide biosynthetic process. It's a polymer which contains uronic acid and N-acetyl glucosamine found in cell wall. Then it is operated by, this is the operand. Now in operand there are many genes, isn't it? So DAUB. So the function of most of the genes are known except for TUFA. This is not, it's not known as yet. Okay, so that is why we are more interested to know what the function of this gene is. From this in silico analysis, we hypothesize that this is the OXDC-like grouping containing domain, which might play a crucial role in acid tolerance mechanism and in maintaining cell wall architecture of the bacillus amyloid. So our in silico data says that this is an OXD containing ornithine carboxyl deoxylase containing protein. It has a grouping domain and it is probably involved with the cell wall biosynthesis. So now we went for biological validation. So what we did, again, we looked at the expression of this gene, a BG01 hypothetical protein, with 2 different conditions. Here, A was pH 7, okay. Then this red one was pH 4.5, which was maintained with hydrochloric acid. This was pH 4.5 again, maintained with acetic acid. And this was soil extraction, okay? Because soil is acidic, we put the soil extraction also to grow. So we grew those bacteria, amylolipifications, in all these 4 different media with different pH maintained through different by different processes. Now, as you know, these organic acids are more harmful to bacteria compared with the inorganic acids. Okay, so you can see in acetic acid what is happening. The expression of this gene is almost 33 there, nearly 33. If you can see, nearly 33-fold higher, whereas as you compared with the normal cell. So it indicates that this protein does play a role in what? Acid tolerance, conferring acid tolerance. Okay. So what we did was we knocked this gene out. Okay, we disrupted the gene in the bacillus amylolipifications. How we disrupted it, we don't go into the detail. If you want, you can talk to Dibbo, because Dr. Dibbo is sitting here. So, like in the plant process, you have agrobacterium. We don't use agrobacterium. We have different vectors for knocking the gene out. So we use that gene. We knock the gene out. Basically, this is showing. See, when you run a gel, you have to have a positive control, a negative control, and your samples. So these are the samples. Okay, this is a negative control, and this is a positive control. Let's see from here. So erythromycin gene was used as a marker. So if the erythromycin gene is there, means it's transformed. So we look for the expression of the erythromycin gene. Okay. And after knocking the gene, we look at the morphology of the bacteria. So you can see this is a normal wild type bacteria. It's it produces a lot of exopolysaccharide. It's sticky. If you grow amylolipifications in a place, you'll find that it has spread and it's sticky in nature. Now when we find the Benedictine one, it's no longer sticky, it's no longer secreting those exopolysaccharides. So there was morphological difference between these two. Now simple morphological difference also will not do. Again, because our interest was with acid tolerance, we saw that what is happening in this. So if you can look at this curve, this is 7, pH 7, okay, pH 4.5. This is cupin mutant, and this red one is cupin mutant, and this is another mutant, 7 at 7, and this is cupin mutant and going at 4.5. So you can clearly see from this experiment that in the mutant, it's not being able to grow at 4.5. No doubt, the wild type also grows slow, but compared now, the mutant is unable to grow. It's much slower as the growing in a normal pH. So it indicates that gene is essential for survival in the acid condition. So this is again a microscope, electron microscope photograph showing the morphology of this. If you can look at the wild type at 6 hours and 12 hours, you see this is the secretion of the exopolysaccharides. Okay, these are the exopolysaccharides secreted by the wild type bacteria. Whereas if you look at the mutant, there is no exopolysaccharide secretions here. Then I mentioned that it produces exopolysaccharide. So we wanted to see, look at this is the mutant growing, and this is the wild type. So wild type is producing this exopolysaccharide you can find in the media, whereas you do not find in this. So okay, we do the experiment again here, biofilm production. This also shows that it is not there. So this is the UPS production was calculated. So in wild type it's more, okay, and Q3 mutant, you don't find in pH 4.5, you don't find any exopolysaccharide secretion at all. So it proves that we were successful in knocking the gene out. Okay, so that was all about how you can use transcritomics, not only to understand how bacteria responds to the stress condition, but also to identify a gene. Okay? So from unknown gene, hypothetical gene, now we know what the gene function of the gene is. So now this has added information in the basic knowledge of the acetomics mechanism. I quickly touch upon this whole genome sequencing of bacteria that we have been doing in our lab. So just give an example of what we took Bacillus altitudinus. The Bacillus altitudinus is known as a plant growth promoting bacteria. Okay, it's an endophyte, plant growth promoting bacteria, and we were interested to know how what is the whole genome of this bacteria, because what genes it might contain. So this is a workflow of the microbial whole genome sequencing. So we had the high quality sequencing, high quality tissue samples. Then this is how this is the wet lab procedure. The wet lab also does some DNA extraction, DNA fragmentation, fragment selection, fragment amplification, and high shotgun sequencing. After that, you have the bioinformatics. This is the genome assembly. This is all the bioinformatics. Again, Norman has done this, so if you have questions, you can ask too. And after this is done in bioinformatics analysis, you have the draft genome. So now we have the draft genome of this bacteria. So this is the comparative genomic map of Bacillus altitudinus strain. So you have this Bacillus our strain. This is the forward. This is the reverse. Okay, all the strands were sequenced. And this one shows the GC content of the our bacteria, and this again shows the sequence of our bacteria forward and reverse. Okay, plus and minus strand, and this compares with the other bacteria, other strains of the bacteria. This colors are different. So you have this is ours. Okay, these two are ours, and these two are all comparing. We are comparing it with the other reference bacterial genomes. This is what we get the map. And from the map, if you look at this, it's a flower pot. Now these 316, these are the core genes which is found in all the bacterial strains that we have analyzed. Okay? And this petals represents the single tone, means unique sequences that you find in the bacteria. So we find that there are 74 unique genes, single tone, in our bacteria. Then from this functional genomic analysis, we looked at, now out of this, you have this subsystem features, what they're like cell wall synthesis, cofactors, potassium metabolism. This pie chart is showing you the genes which are involved in different processes. Okay? Now out of this subsystem coverage shows that till now, 30% or 38%, 30% of these genes have been annotated. So we know what the function of these genes are. But 70% remains to be annotated. We still do not know. And hopefully, as the database gets updated, we'll also come to know what this function is. Okay? Or otherwise, again, we have to go the procedure that we applied, looking at the function, knocking it out, and seeing the genes one by one. Okay? So this is a pie chart showing the distribution of genes involved in plant interaction and plant growth promotion. So these are some of these things which shows how this, what are the different genes that are involved with the plant interaction and with the plant growth promotion. So if you look at here, this about 26.09% is involved with, establishing relationship with the plant. So this is And then here is in silico identification of genetic factors involved in plant microbial culture. If you look into this closely, you'll find that these are the genes, okay, which are involved. These are the features, and these are the genes involved in these features, which are present in the genome of this bacillus altitudinus. And this Bacillus altitudinus, although we were looking for plant growth promotion activities, we find now, looking at the whole genome, we find that it also codes for some secondary metabolites. And some of the secondary metabolites could be related with the plant protection mechanism as well. So maybe this, we have not tested till now, but maybe this bacteria also, looking at the whole genome, maybe this bacteria also has a biocontrol activity. Okay? So this is all, and this all is work of the lab together. Many of them are sitting here today. So it's all thanks to them that, I'm here today. So thank you all very much. If there are any questions, I'll be happy to answer. No questions. Was I so clear that you don't have any questions?
Online participants can also ask.
Huh?
Online participants.
Oh, that was okay.
I think online they are. I don't know whether they are working on microbiology or not. Not, they can ask. I think today they all are very much tired.
Hectic
Very
days.
hectic days. Less sleep, too much of work, too much of laboratory work, right?
Do they have laboratory work today afternoon or is it over?
Afternoon they have a little bit, I think Bippo has to show the cloning part.
Do you have something people planned today? Now?
And they are very much happy to see microbiology work. So I'm sure they will be happy to. Huh? Cloning on.
Oh, cloning on. Cloning on. Okay then. If you have any questions, I've already told the contact person. Please talk to them. They are here with you all the time. You can ask. Okay.
You have mentioned about HAS, right?
About what? Sorry.
Us.
Oh yes, yes.
So in that, what one sample you do, or what price sample you do? Like,
Okay. No. See.